work in the same lab as Chris, and we use the same kind of genetic techniques to label different cells, but I'm looking at very different cells and a very different aspect of those cells. I'm looking at the movement of mitochondria in astrocytes. So let's start off by setting the stage a little bit. So if we look at humans or dogs or pretty much any animal, we all have, we all have brains, which is good news if you're a neuroscientist and actually like to look at these things. And what's interesting is while these, obviously in all of these animals, the brains are very different sizes, very different shapes, the cells that make up these cells, these brains, are actually remarkably similar, function in remarkably similar ways. And that's why we think that by studying the cells in, for example, the brain of a mouse, we can not only learn something that's interesting about that mouse, but actually something that applies to us to understanding our brains and hopefully to curing the diseases in humans. So if we take a closer look, there are a lot of different types of cells that make up the brain. We've already heard about the neurons. These are the cells that usually get the most attention to have all these electrical activity patterns. But there are a lot of other cells, and one of the primary other cell types, and the one that I'm interested in are here in the middle, they're called astrocytes. And while the astrocytes don't have electrical activities, they do show these remarkable patterns of calcium fluxes, which we can visualize in live cells under the microscope. And we don't have nearly enough time to go into what these signals are all about, and there's a lot of debate in the scientific community about the function of these calcium patterns. But what I'm trying to show here is that just because they're not electrically active, it doesn't mean that they're inactive. They still do a lot of stuff. And this is actually very much of what I do in the lab. I take these kinds of images and these types of movies, and then I'm trying to see that what happens if I'm manipulating one of these astrocytes. How do their activity patterns change, and even more interestingly, how do their interactions with the neurons around them change? Now, if we look even closer, we can start looking at the individual parts that make up the cell, in this case the astrocyte, and the machine that I'm interested in is called, these little green bits in here, which are called mitochondria. They're often referred to as the power plants of the cell, even though they do a lot more, but that's not important for now. And to understand the mitochondria and to understand their role in the cell, I like to compare it to the analogy of comparing a cell to a city. A city has all of these different parts that are connected by large and small streets, small highways, transport highways. And what I think actually the analogy to the cell holds up really well is that a city has some rules that guide the entire city, but the individual parts, the individual people kind of in a day-to-day life do their own thing, yet still somehow, even without being aware of each other, work together to run the entire entity, run the entire city. And that's very much how I like to think about the individual parts of a cell. And what I'm interested in is how the cell powers itself. And if we stick with the city analogy, if we want to read at night in Montreal, we usually start by building hydroelectric dams up north, and then have large power lines bringing electricity down from the power plants to the city, and we can turn on the light. And now if we go back to one of the cells that I showed earlier, as I said, the cell has something that functions very analogous to a power plant, which are the little green bits, which are the mitochondria, but the cell doesn't have anything that's analogous to the power lines. So the cell can't actually transport energy from the mitochondria to where the energy is needed, and instead has to transport the entire mitochondria. So for that, there are molecular machines that function very much like a train, actually physically relocating the mitochondria, pulling them along tracks called microtubules. And this is the process that I'm actually interested in. This is the process that I'm trying to study. And by study and understand, a molecular biologist like myself means I'm trying to break it. So I have developed molecular tools to actually, if we stick with this analogy, take the wheels off the train, and now I'm trying to see what happens to the cell and to the cells around, to the surroundings of these astrocytes. And this is just a small contribution trying to understand this one small mechanism in the cell, but I think it contributes ultimately to a better understanding of how this entire cell functions, and then further how this cell functions in the context, functions together, interacts with other cells, which ultimately leads to better understanding of the brain as a whole.